Hello everybody, my name is Matheus de Paula Corrêa, and currently I'm a Master Science student at the Molecular Biology Postgraduate Program of the Federal University of Brasília. I'm working on the analysis of sRNA-Seq data originated from root samples of Arachis duranensis plants subject to cross-stress conditions, more specifically biotic stress, infection of root cells by Meloidogyne arenaria root knot nematodes, and dissociation of root knot nematode infection with drought stress. As a brief introduction for why this research line is important, population growth and climate change are important challenges faced by humankind in the 21st century. It is predicted that the frequency of extreme climatic events, as well as the occurrence of desertification and salinization, will increase, subjecting crop species to a broad range of biotic and abiotic stress factors. Two of the main stress components predicted to influence crop growth and development in the near future are drought stress and root knot nematode parasitism. MicroRNAs are deeply involved in the regulation of plant responses to biotic and abiotic stress conditions. These nucleic acid species originate from the transcription of miR genes by Bowtie with subsequent processing and cleavage of double-stranded transcripts by a set of nuclear proteins, which yields a varied quantity of microRNA star duplexes. These duplexes are then transported to the cytoplasm, whereby one RNA strand will be selected and incorporated into a protein complex called RISC, which will exert post-transcriptional regulation of gene expression through translation inhibition or cleavage of the target messenger RNA transcript. In this research, we used root samples from Arachis duranensis plants, which constitute a suitable model to study plant responses to cross-stress conditions due to its inherent tolerance to drought and resistance to root knot nematode infections. Therefore, the main objectives of this research are: study the microRNA diversity in samples of root tissue of Arachis duranensis plants exposed to cross-stress conditions, ID express microRNAs, annotate express microRNAs, quantify and analyze differential expression of microRNAs. The samples were sequenced through the Illumina sRNA-Seq protocol. The research is composed by 3 treatments: control, labeled as CTR, samples inoculated with Meloidogyne arenaria, labeled as INOC, and association of root knot nematode inoculation with drought stress, labeled as STR. The analysis pipeline used for this research was developed by LBI, Bioinformatics Laboratory at Embrapa Recursos Genéticos e Biotecnologia, and can be divided into 5 main steps. Step 1, pre-processing. Step 2, ShortStack clustering. ShortStack is a software tool that uses a set of standardized criteria to categorize and annotate plant microRNA genes, and thus it has been used in the present research. In step 2, the processed data were analyzed through ShortStack, which generated as output the identified clusters with information regarding read counts, precursor prediction, sequence prediction, and categorization of the prediction according to the standardized criteria. Step 3, miRNA. As a source of alternative cluster predictions, the processed data were also analyzed through miRNA, a sRNA DeepSeq analysis software tool that maps DeepSeq reads to reference genome and also predicts the secondary structures of each mapped locus. Step 4, ShortStack with miRNA clustering. In step 4, ShortStack was run with the information of the loci predicted by miRNA by means of an input text file. Step 5, annotation. Each of the microRNA sequences predicted by step 2 and step 4 were compared with already annotated sequences from miRBase, a searchable database of published microRNA sequences and annotation through BLASTAN, with the objective of identifying conserved microRNA sequences. The unannotated microRNA sequences were then manually created through secondary structure analysis with struct-based software. As preliminary results, 143 putative microRNAs from 29 miR families were identified. From these, 24 putative microRNAs may be novel due to lack of annotation in microRNA databases, large number of reads, secondary structure adhering to plant microRNA annotation criteria, such as these 2 sequences whose secondary structures are being displayed. Next steps of the research include microRNA differential expression analysis and microRNA target prediction. Thank you very much.